Hey guys, welcome back to my channel. Today I am here with some previous year questions for microbiology, which is a part of your GATE Life Sciences exam. So if you're preparing for this exam, then please do subscribe to my channel because I would be uploading videos related to previous year questions and some other things which are related to this exam on my channel every day. So for the last some days I was not uploading any videos, but now I would be regularly uploading videos for the GATE previous year questions specially. So stay tuned to my channel and please do subscribe and share my videos with your friends who are also preparing for this exam. So let us start with this video. So the first question is, David Baltimore's classification of viruses is based on differences in: first, host cell receptors used by the viruses; B is the pathways required to synthesize virus mRNA; C is the modes of transmission of viruses; and D is the envelope proteins on the surface of viruses. So these are some things that have been told that what is the basis of classification, किस basis पर viruses को classify किया गया है. So the correct answer is the pathways required to synthesize virus mRNA is the basis of the classification which David Baltimore has done. Which of the following eukaryotic cellular components carries out intracellular degradation during autophagy? So first we should talk about what is autophagy so autophagy is basically a phenomena that is used for cleaning out damaged cells from the body. Okay, the body is cleaning itself. It is removing out the damaged cells. So it is doing this to regenerate newer and healthier cells. So this is what autophagy is, and we have to tell in which organelle it is being carried out. So you have different organelles within a cell. You have nucleus, you have Golgi bodies, you have ribosomes, you have lysosomes. So if you have studied about the organelles, then you must be familiar with the hydrolytic enzymes, which organelle consists of hydrolytic or degrading enzymes. So lysosome consists of degrading enzymes or hydrolytic enzymes like nucleases, and there are many other, okay, proteases, nucleases, lipases. Okay, so these all lytic enzymes are found in the lysosomes. So lysosomes is the only cellular component in the eukaryotes which carries out intracellular degradation during autophagy. So the option D is the correct answer. Now we have a match the following, in which we have been given some microorganisms and we have to tell the mode of transmission. So what is the mode of transmission? The mode of transmission of the disease. Okay, so first one is Bordetella pertussis. So its mode of transmission is droplet infection. This is done, this transmits through droplets. Okay, dengue virus, you must be knowing about it. It is vector borne. It needs a vector for itself. Entamoeba histolytica is caused due to the contaminated food. When the person consumes contaminated food, the food which is contaminated with entamoeba, then the person gets the disease. And hepatitis B virus is being transmitted through the blood. So it is bloodborne. So the option A is the correct answer, which is showing all the correct match the following options. Now analysis of DNA sequences suggests that eukaryotic mitochondrial genome primarily originated from, when we are doing, so this question basically says that when we are analyzing the DNA sequences, then we get to know that the eukaryotic mitochondrial genomes which are found in the cells are primarily originated from which of the following organisms. So we have fungi, we have protozoa, we have algae and bacteria. So basically the eukaryotic mitochondrial genomes primarily originated from the bacteria. Okay, they were first found in the bacteria. Binomial nomenclature has not yet been adopted for. So binomial nomenclature has been adopted for bacteria, fungi, and it has also been adopted for protozoa but not for the virus. So the option C is the correct answer, that the binomial nomenclature has not yet been adopted for the viruses. Which of the following is not an accepted method for sterilization? You have autoclaving. You know very well about the autoclaving. It is very well-known method for sterilization. X-rays are also involved. Gamma rays are also involved. But the UV rays are not involved in the methods of sterilization because they are of a very damaging nature. they damage the DNA and RNA of the organism. That is why UV rays are not used. Now we have the match of the following again, in which we have antimicrobial agents and we have the category or mode of action. So we have to match them. So fluoroquinolones are having the mode of action which is inhibition of nucleic acid synthesis. Okay, they inhibit the nucleic acid synthesis. Then we have amphotericin B. Okay, so amphotericin B is basically having it is a antifungal agent. Okay, so it retards the growth of fungus or any fungi that is being growing. Then we have tetracycline. Tetracycline inhibits the protein synthesis. Okay, so that is the option Q. Then amoxicillin, it is inhibiting the beta lactam antimicrobial. It is having the antimicrobial tendency and it inhibits the beta lactam synthesis. So the option C over here is the correct answer. Which of the following is terminal electron acceptor in the given metabolic reaction catalyzed by the methanogens? So we are having this reaction which is being catalyzed by the methanogens. So in this we have H2, CO2, CH4 or H2O. We have to tell which is a terminal electron acceptor. So carbon dioxide acts as a terminal electron acceptor, whereas H2 acts as a terminal electron donor. So I will write it over here. This is a donor and this is a acceptor in this reaction. Okay, so you must know both of them. Anyone can be asked. So as in here, terminal electron acceptor has been asked, which is the option B, carbon dioxide. Now the primary product of nitrogen fixation is, I hope you must have studied nitrogen fixation since the class 12. You must be very well familiar with this particular topic, and this is a very important topic that has been asked several times in your GATE exams. Almost every year this question has been asked, means questions from this topic have been asked. So you have to tell that dinitrogen or ammonia or nitrogen dioxide or nitrate, which one of the following is the primary product of nitrogen fixation. So ammonia is the primary product of nitrogen fixation, which is the option B over here. Now in humans, the key stages in the life cycle of malarial parasite occurs in red blood cells and the liver, red blood cells and platelets, red blood cells and the pancreas, or red blood cells and the gut. So if you have studied about malaria, then you must be knowing that the key stages in the life cycle of malarial parasite occurs in the red blood cells and the liver cells or the liver. Okay, so this is the correct answer over here. Now we have a match the following again, where we have precursors or intermediates and we have to tell their metabolic pathway, or we have to tell from which metabolic pathway they are being associated to. So we have inosine monophosphate, which is being associated with the purine biosynthesis. So R is the purine biosynthesis. Then we have ornithine, which is associated with the S-arginine biosynthesis, which is the option S. Then we have Corey's mate, which is associated with L-tryptophan biosynthesis, and homocysteine is associated with L-methionine biosynthesis. So the option D is the correct answer over here. Now we have one more question for this video. This is the last question of this video. Accumulating evidence suggests that domain archaea is more closely related to domain eukarya. तो इस क्वेश्चन में से आपको काफ़ी सारी चीज़ें पता चलेंगी. पहली चीज़ ये है कि डोमेन आर्किया जो है डोमेन यूकैरिया के साथ ज़्यादा रिलेशन रखती है एज कम्पेयर टू डोमेन बैक्टीरिया. ओके, सो डोमेन आर्किया is more related to the domain eukarya. Okay, which of the following properties are shared between eukaryotes and the archaea? So we have to tell which of the following properties are being shared by both of them. Is it protein biogenesis, presence of sterol containing membranes, ribosomal subunit structures, adaptation to extreme environmental conditions, fatty acid with ester linkages in the cell membrane? So they share protein biogenesis property as well as ribosomal subunit structure. So the option C is the correct answer. So only these two properties are being shared by the domain eukarya and the domain archaea. Okay, so that's all for this video. So if you guys need any other help except these previous year questions, you are free to contact. If you want me to make more videos related to the gate preparation, then please let me know. And please do like this video, share this video with your friends, show some support so that I can make more such videos. I can be motivated to make more such videos. And please subscribe to my channel, because if there won't be any subscribers, there won't be anybody watching my videos, there won't be any use of making these videos. So please do subscribe and share these videos with your friends so that I could be motivated and to make more such videos, so that you can be getting some help through these efforts to clear your gate exams. And I hope that most of you who are watching my videos will obviously clear the gate exam. So please do subscribe and stay tuned to my channel. Thank you very much for watching.